Welcome, everyone. Good afternoon. I'm Eva. I'm a mathematical modeler here at ECDC, and I'll be moderating today's talk. And we're very happy to welcome Stephen Kissler, who's an assistant professor of computer science at the University of Colorado. And will give a very interesting talk on integrating viral kinetics into routine outbreak surveillance. I'm very excited to hear what he has to say. A little bit of housekeeping. The talk will be 30 minutes, and we'll have enough time for questions and discussions afterwards. But if you have any pressing questions, raise your hand or put them in the chat even during the talk. Be civil, but yeah, if you have any questions, just raise them. And that's all I have to say. I'll hand over to Stephen. Thank you so much for joining us.
Thank you.
And maybe if you're not talking, if you can mute yourself, that would be great. Over to you.
Great. Thank you very much. Okay. Looks like we are live. Cool. Well, thank you so much, Eva, and all the rest of you, Renee, Rock, for organizing this and for inviting me along to speak this morning. I'm really excited to share some of these ideas and really to get some feedback from you all on some of these things that we've been thinking about recently. I've focused quite a bit on viral kinetics over the past couple of years since the start of the COVID-19 pandemic, along with many of my colleagues. And one of the big questions we have right now is what we do with this going forward. So I'm going to be talking today about how we might integrate viral kinetics into routine outbreak surveillance going forward, focusing on some of the things that we've learned during the COVID-19 pandemic and specifically highlighting some of the challenges and opportunities that we see. But of course, this is not going to be an exhaustive list, and I would love to hear from you both about the challenges and opportunities that you see in this area too. This work has been sort of a large collaborative effort. We have an article that is going to describe some of the things that I'm going to be talking about today that should be forthcoming, Philosophical Transactions B. And so just a screenshot of the proof of that, which hopefully should be out in just the next couple of weeks. But this work has been co-led by James Hay, Dan Larimore, and Aishania Tresh, whom many of you, I think, may also know. And so I'd like to begin by just making sure that we're on the same page when thinking about viral kinetics and what we mean by them. Because actually viral kinetics can refer to a lot of different things, and there can be a lot of useful ways of measuring them. For most of the talk, I'm going to be sort of collapsing and simplifying what we mean by viral kinetics, but I think it's worth keeping some of these other elements of viral kinetics in mind. So of course, viral kinetics could refer to anything that has to do with the growth, decay, and movement of virus. And that can happen in lots of different types of compartments, both in the environment. So we could think about virus in sort of ambient air. We could think about the kinetics of infection. And when we're thinking about the kinetics of infection, we often have to think about what's happening in different anatomical sites, whether we're thinking about the nose or the throat or the lungs. We could also think about viral kinetics of exhaled virus in breath, which might be really important if we're thinking about infectiousness. And when we're doing all of those things, we need to think about not just viral growth, but also viral clearance. So most of the data that I'm going to be showing today from COVID-19 and most of the sort of canonical examples that I'm going to be bringing up are going to be specifically thinking about acute respiratory viruses as measured in the nose or the throat through nasal swabs or things like saliva. So, we're taking a swab. We might be serially sampling someone over time. And when we do that, we can get a plot of viral load over time. And so viral load goes up until sort of the peak of infection and then comes back down. We often measure it on a logarithmic scale. And so if you think of viral growth increasing exponentially and then decaying exponentially, potentially a slower rate, log transform that on the vertical scale and you get sort of these triangle looking things. I don't want to restrict our discussion to just acute respiratory viruses. I think a lot of the things that I'm going to talk about can apply to bacteria and parasites as well, along with things that are potentially more chronic. But this is going to be sort of the example that I'm going to be keeping in mind while we're sort of very open to extending this to other things too. Okay, so why might we care? So when we're thinking about especially an emerging infectious disease, but also infectious diseases that we commonly encounter, there are sort of two scales of things that we often think about. One of them is the host pathogen interface, where we might be interested in things like the immune response or the phenotype of the virus itself and how it behaves in different contexts. Or we might be interested in how infection plays out in different tissues in the body, like the lungs. But on the other hand, many of us on the call are epidemiologists, and so we're interested in what happens at the population level. So we might be interested in understanding how people develop symptoms and how likely it is that a person develops symptoms. And we might be interested in understanding the outcomes of diagnostic tests so that we can use those to better control an epidemic. And ultimately, we want to fold these things into infectious disease models, so that we can get some sense of what the epidemic is going to do as it unfolds. Viral kinetics, I think, occupy this really useful space. They are, I think, one of the most critical links between these two different scales, because they are the emergent property of those within host dynamics, but then they also are sort of the foundation of the epidemiological dynamics. And so as a result, if we can understand something about viral kinetics and how they change across pathogen variants and how they change across contexts, we can sort of move in either direction, and we can move up to the epidemiological scale or down to the within host scale, and even sometimes translate between them using viral kinetics as this mediator. So they can potentially be a really powerful source of information. And over the next couple of slides, I want to talk a little bit about more specifically the type of information that we might be able to glean from viral kinetics and that we have gotten from them, both during the COVID-19 pandemic and then also with other outbreaks of infectious diseases. So I want to split this next section up into thinking about basic epidemiology, clinical medicine, and designing outbreak interventions. So in other words, thinking about sort of what happens in an uncontrolled and sort of unintervened upon epidemic, and then what happens when we try to use viral kinetics to help us with individual patients, and then what happens when we use viral kinetics to help us understand what to do at the population level to control disease. I think one of the key applications of viral kinetics is giving us information about the timing of infectiousness. And this can be really critical for just understanding how quickly an outbreak is going to spread. I'm going to be using these diagrams, these timelines of infections over the next couple of slides. So basically depicting an infectious exposure or an infection that happens with this black X. And there may be a period where a person is not yet infectious, and then at some point they turn infectious for some period of time in red. And then symptoms may happen after the start of infectiousness or before, and they may linger longer than infectiousness or end before infectiousness ends. So there's usually a correlation between symptoms and infectiousness, but there can be a lot of variability in the timing of the onset and the offset of those things. So when we're thinking about basic epidemiology, the details of this timeline end up really mattering. And to parameterize things like infectious disease transmission models, we might be interested in things like the incubation period, because that can let us back calculate from the onset of symptoms to the timing of exposure. And critically, we also need to know something about the generation interval, which is the time between a primary infection and then a secondary infection that it causes. And if we don't have a notion of viral kinetics, but maybe we're doing some kind of household study, we might sample people and identify positive tests, and so that we know that these people were infected at some point in the past. But we have to do a lot of inference and statistics to try to understand exactly when those infections first occurred. And this is one of the ways where an understanding of viral kinetics can begin to help us. So if we instead envision a world in which we've maybe taken a couple of tests from these people, and these tests are maybe quantitative, then if we have an understanding of how viral kinetics play out over time, we can fit a curve to those data, and we can much more accurately back calculate the timing of the onset of infection. So that's one area where by fitting viral kinetics models, we can get a much better understanding of the timeline of infection, of the natural history, really, of infection. And by doing that, we can get a lot better estimates of sort of the basic epidemiology of a pathogen. We can also get other things potentially for free or roughly for free from this kind of data. So if we have an understanding of how much virus a person needs to produce to be infectious, of course, measuring this is still a pretty open challenge for many different pathogens. But if we can have some rough sense of when a person might be infectious or not based on how much virus they're producing, that can also help us to parameterize the infectiousness period or how long a person remains infectious. And that can also be really useful for parameterizing outbreak models. If we take that idea one step further, we can also get a sense of how variable the sort of inherent infectiousness of people is across a population. So on the right, I'm depicting some actual data from a study we worked on during the COVID-19 pandemic. So each point represents the outcome of a quantitative PCR test, and on the vertical axis is viral load as measured by the cycle threshold from that test. And there's a huge amount of variability across people. If you fit lines to the individual kinetics trajectories of different people, you see a lot of different types of trajectories emerging. So on average, a person has their virus grow at some rate and decay at some rate and sort of reaches this middle level of viral load. But there are some people who have these really high peaks, and some people who have very slow clearances. And you might expect that these infections might play a really important role in transmission because of the intensity of infectiousness or because of the duration. So if we can measure viral kinetics and we can understand sort of how they vary across people, then we might start to get some insights about the propensity for super spreading for a given pathogen as well. Of course, super spreading deals not just with biological infectiousness, but there are also a lot of social factors. But if we can at least isolate some of the biological factors, then that can at least start to give us some indication of to what degree super spreading might be a factor in a given infectious disease. In the context of clinical medicine, viral kinetics can also be useful. One of the areas that understanding viral kinetics can help is to guide the administration of therapeutics to patients who might have been infected. So for a lot of antivirals, including things like Tamiflu and Paxlovid, there's a specific administration window, and you really want to make sure that you're giving that antiviral early in the course of infection. After the infection has sort of gone on for a certain period of time, that therapeutic becomes less and less effective. And so oftentimes the treatment algorithms for these therapeutics are often in terms of the onset of symptoms. So you want to make sure that you give something like Tamiflu within a short period of time after symptoms have developed. And otherwise, you can measure it in terms of the time after the first positive test. But we might be able to get more information, again, from quantitative tests. So if you have a positive test and you know how much virus was being produced at that point in time, you can either use that to sort of explicitly guide whether the person is early enough in infection to administer the therapeutic, or again, you can fit some kind of viral kinetic model, back calculate when the infection began, and use this to have a better sense of when it might or might not be appropriate to administer a given therapeutic. And then at a broader scale, we can also use viral kinetics to integrate them into vaccine and clinical into trials for vaccines and therapeutics to sort of provide us with alternative endpoints to just the sort of standard endpoint of whether or not an infection happens or how long an infection lasts. So this was a really exciting area of work that is not really novel, but I think gained a lot more traction during the COVID-19 pandemic, where, for example, during a vaccine trial, people would be serially tested, and they would be serially tested with quantitative PCR. And so if a person did become infected and they continued to be tested over the course of their infection, we could still see whether that person produced less virus over the course of their infection on average, or if they cleared it more quickly. So basically comparing the solid versus the dashed lines. And so this can give us both basically alternative endpoints for these trials, different ways of demonstrating whether or not a therapeutic or a vaccine might be effective. But they can also help to give us some clues as to how these therapeutics and vaccines might behave when we deploy them at the population scale. Because again, if even if these things are not necessarily completely preventing a huge number of infections, providing sterilizing immunity, the immunity that they do provide might make a person less infectious or might modulate sort of the window of time when a person is infectious. And that can be really useful to know when we're trying to understand sort of what the impact of these things are going to be when we deploy them broadly. And then the last thing, one of the other main areas where I think viral kinetics can be really valuable is when using diagnostic tests as a tool for public health response. So we can think about adaptive non-pharmaceutical interventions. We can use diagnostic testing to inform a lot of different things, like how long a person should isolate, or to do things like screening to detect people before their infections peak and before they cause a lot of infections. You can imagine that these different public health applications, whether we're thinking about isolation and the duration of isolation or isolating people early, might require different strategies for how we test people, how frequently we test them, sort of the span of time between tests and between positive and negative tests, and having an understanding of sort of the baseline viral kinetics across the population can help us design efficient and effective testing-based non-pharmaceutical interventions. And then last, this is sort of a broader scale application of viral kinetics. We can use cross-sectional measurements of viral load in a population to give us a sense of where we are on an epidemic curve. This is work that was really led by my colleague James Hay and others. But the idea is that basically, if you do cross-sectional viral load measurements across a population early in an outbreak, on average, the people who are infected are going to be early in their infection. And that might shift viral load measurements a little bit higher on average. Whereas at the end of an epidemic, infections are going to be on average relatively old, and that might shift them relatively lower. So it's almost like this like red shift, blue shift kind of phenomenon that you get, but an epidemiological one. And so if you can compare cross-sectional viral load measurements over time, you can actually get a pretty good sense of where you are on an epidemic curve and whether the epidemic is growing or decaying. And that, again, can be really helpful for providing sort of public health guidance and just understanding where we are in an outbreak. Okay, so hopefully I've made the case that measuring viral kinetics and understanding something about them can be a really useful thing to do during infectious disease outbreaks. As a brief aside, I want to talk a little bit about modeling because I think there is an important question as to why and whether viral kinetics models are important or whether we can just do what we need to do with quantitative viral load measurements over the course of an infection. I am biased. I am a modeler. I do think that models are really important. But I think that there's a clear case to be made for these things because if we think of a viral kinetic model as essentially just a theoretical curve that describes a person's sort of actual viral load over the course of their infection, then applying a model to the data that we gather allows us to extract a lot more value out of them. So rather than just having sort of the course of viral positivity over time, we can really start to extrapolate to different parts of the infection. We can start to make inferences about how viral kinetics differ across different demographics and these kinds of things. And so having some kind of mathematical characterization of viral kinetics that underlies the data that we're collecting can be a really useful thing to have. And can be critical for gaining some of the insights that are necessary for some of those basic epidemiological applications, the clinical medicine applications, and the public health applications. There are a couple of different sort of main modeling paradigms that we use when thinking about viral kinetics. And I usually refer to these as mechanistic or phenomenological. So we'll talk briefly about those and the sort of the merits and pitfalls of them. Mechanistic viral kinetics models are sort of the older and more established version of viral kinetics modeling. And the idea here is really that we want to mathematically describe what's going on inside the body over the course of an infection and to translate immune dynamics and viral abundance into specific explicit mathematical equations. And so we might take diagrams like these that represent things like target cells and exposed target cells and the number of infectious virions in the body, and then translate those into ordinary differential equations. And then we can fit those equations to data, and we can get curves out. And that could be a really useful thing, especially if we're interested in what's going on at the within host level. On the other hand, there are also these phenomenological models where we sort of ignore all of this rich detail of what's happening inside the body. And instead, we just impose a shape on our viral kinetic trajectories. One of the most common ones right now is simply one of these sort of triangular curves. And so here we might have 3 different control points. These are basically just hinges on a piecewise linear curve that we can move back and forth and up and down. And our goal is to just find where those hinge points best match the data that we have. There are a lot of sort of different things that we might say about these types of models. So again, mechanistic models can give us really nice insights about immunology, and we can use them to predict viral kinetic features. So for example, these types of models were really useful for sort of postulating the existence of rebound infections in COVID-19. Because when we fit the models, and by we, I mean sort of the field, I was not fitting these mechanistic models to COVID-19 data. But when you fit them, for some values of the parameters and in some contexts, you can solve the differential equations. And after the initial infection peak, you actually get another peak later in time. And so you could hypothesize the existence of rebound infections before they were even sort of clinically observed, which was really interesting. It's not something you can do with a phenomenological model very easily. But of course, mechanistic models can be hard to fit, can be hard to specify the parameters. Sometimes you need to specify things like the number of epithelial cells in the respiratory tract, and those can be really uncertain. And so model selection can also be really challenging. Phenomenological models, on the other hand, often are really useful epidemiologically, so they can tell us sort of how viral kinetics differ across population groups. They give us really robust uncertainty measurements, and they can be quick and straightforward to fit, even if we have large data sets. So that's becoming more and more relevant. But of course, they provide less information about what's happening within the host. And sometimes the structure that they impose on viral kinetics can be too rigid, and it causes us to potentially overlook really important features of the viral kinetics curve that we might want to be able to account for. So I've laid out sort of the benefit of viral kinetics modeling very briefly, or really the benefit of doing surveillance and understanding something about viral kinetics. And then thinking about models as a way of sort of gathering more information from the viral kinetics data that we collect. And so now what I want to do in these last few minutes is to talk a little bit about what happened with viral kinetics during the COVID-19 pandemic, and even talking a little bit about what happened before. And then to sort of actively think with you all about what we might want from viral kinetics surveillance and modeling going forward. I'll go very briefly through some of these next slides. So viral kinetics modeling did not begin during the COVID-19 pandemic. Scientists have been thinking about viral kinetics since at least the 1940s with early studies on viral, basically bacteriophage growth. But it really wasn't until the HIV pandemic when we really started thinking about viral kinetics within the human body. And a lot of that was spurred both by the availability of quantitative PCR and then also by the development of some of these mechanistic mathematical models that allowed us to start to make sense of this really complex natural history of HIV infections. Between the 1980s and the more recent history, there was definitely viral kinetics work going on in various other infections. So some of these mechanistic models were starting to be applied to things like influenza A and MERS, and our data collection strategies were being refined more and more. But it was really the COVID-19 pandemic that I think produced another step change in our approach to understanding and thinking about viral kinetics. Part of that was this understanding that we could use diagnostic tests in a potentially nuanced way to help control the spread of the outbreak. So how do we use testing to make sure that people isolate when they need to and isolate for as long as they need to? But the real thing was data collection. So we had people getting quantitative PCR tests all the time in a lot of different contexts. We had this happening at universities to try to control spread. We had this happening in sort of robust population-wide studies, like this one that was led by ONS in the UK. And we had it happening in occupational health settings, sort of professional settings, like the National Basketball Association or the Premier League in the UK, where basically a lot of testing was happening so that these organizations could make sure that infection wasn't going to spread. So to talk about those in a little bit more detail, the data that we got on COVID-19 viral kinetics really came from sort of three main places, and these sort of emerged over the course of the pandemic. Early on, a lot of our information came from these targeted studies of hospitalized patients, where a person might present with symptoms, usually very severe symptoms, and they would just be tested over time. And so you can get these viral decay curves that capture sort of how long it takes the virus to be cleared from their bodies. And this was really useful information early on in the pandemic, but of course it misses some of the critical elements of what was happening early in their infection. And a lot of that information is important for understanding how the disease might spread. Sort of on the other side of the spectrum are these large prospective studies. This is a screen grab from a paper that's describing data from the ATTACK study. They're the assessment of transmission and contagiousness of COVID-19 in contacts. This was a great, super well-powered household study that was looking at understanding transmission of COVID-19, but there was also a lot of serial testing of viral load in people who had been exposed to COVID-19. And from that, we get these really nice viral kinetic trajectories. And sort of sitting in between these are these occupational health studies where the data weren't being collected specifically for scientific purposes. And so there are a lot of biases and sort of lack of control, but you get a huge volume of data out because, again, people are being tested super frequently to try to control infection. And from those data, we can potentially pull out some interesting things about the viral kinetics trajectories. So again, when we're thinking about moving forward and thinking about continuous viral kinetics surveillance, each of these types of studies has some sort of merits and pitfalls that we're going to want to keep in mind that have to do with things like how costly and complex they are to deploy, what the sample size are, how representative they are, and sort of how sustainable they are. I think sustainability is a really big question here. So that's one of the big challenges with occupational health programs is that as soon as the occupational health needs, as soon as the NBA no longer wants to collect data on COVID-19, the data stream stops. And so thinking about how we can take these ideas, but make them so that they are part of more sustained efforts is a major concern and sort of something we're thinking a lot about going forward. Okay, so where does this leave us? So one of the things that we're trying to envision right now, and that we would love sort of input and help with, is this question of how we might design a continuous monitoring system for the kinetics of endemic and emerging pathogens. The idea is that this would complement active surveillance efforts. So it would go alongside the surveillance that we're already doing on hospitalizations, mortality, serology, and genomics. And in addition to just the surveillance, ideally we would be able to integrate it into dashboards and situation reports alongside the estimates that we already have of things like prevalence and similar metrics. So we've gotten a lot of inspiration from existing efforts. So things like the NCBI, which allows scientists from around the world to upload information on pathogen genomes. So it streamlines data sharing. It provides sort of a standardized way of sharing genomic information. And that can be a really useful thing to have. And we wonder if there might be something similar that we could do for viral kinetics. Also want to highlight the UK HSA data dashboard. So here is essentially a space where infectious disease surveillance data can be very easily depicted across many different types of pathogens. And this can be really useful for basic scientists, for epidemiologists, and for the general public to get a sense of what's going on with each of these different types of pathogens. So we're trying to think about like what kinds of things might we want to glean from these existing projects to build something similar for viral kinetics. To do that, there are a number of challenges, and I'm just going to go briefly through some of the key challenges that we've identified so far. The first of those is, how do we design viral kinetics studies that are representative, that are well-powered, that are sustainable, and that give us the information that we need at different stages of an epidemic? So this is a table that I'm not going to go through in full detail. This is from the paper that I had mentioned on that second slide. But just noting that what we need from viral kinetics surveillance might differ by stage of the outbreak. So early on, with the first few hundred cases, we might really be trying to parameterize epidemiological models and get a sense of, roughly speaking, how does this pathogen compare to others that we already know about? Later in an epidemic, we might want much larger studies that are giving us a sense of how viral kinetics differ by pathogen variant and demographic group. And then late in the epidemic, we might have different priorities where we're trying to understand sort of how viral kinetics responds to whether or not a person is vaccinated or whether they've gotten treated. And so we really need sort of off-the-shelf study designs that we can use in each of these contexts to collect data efficiently and to do it in a way that that's sort of well-powered for the questions that we have in mind. And so the study design for this kind of work is a major open question right now. Then once the data is collected, we need a place to put it. So right now we're working on sort of building one of these viral kinetics libraries, again, inspired by things like the WHO Global Repository of Epidemic Parameters, which again is just a place where epidemic parameters across pathogens are stored and made available. The Global Vaccine Library, and also things like Next Strain, which aren't necessarily places where genomic data go. Next Strain pulls on genomic data from a lot of different sources, but they also provide a way of looking at phylogenetic models in a really flexible and intuitive and accessible way. And so we wonder if there might be a way to think about incorporating different types of viral kinetics models so that we can grab data from many different sources and start to get a very rapid and intuitive sense of how viral kinetics play out for different pathogens and across different studies. So this is going to be, I think, also a major effort to try to understand how we do this and how we balance the need for data standardization and detail in these very diverse types of data sets that we might get. That points towards a third challenge, which is data reporting and sharing standards. This goes without saying, but of course, the people who are collecting the data and the people who are cleaning the data and modeling the data need to be acknowledged for those efforts. And we also need very clear standards for how the data are going to be reported and interpreted. There's actually already been a lot of work done on this at the national scale. So for example, there's an example from Belgium thinking about how do we interpret and communicate the outcomes of quantitative diagnostic tests. Also some work from South Africa thinking about how do we design these sort of data analysis pipelines for quantitative testing data. Whatever ends up happening, we need to make sure that they are following these fair principles of making sure that the data is findable and accessible and interoperable and reusable. And so figuring out how to do this specifically with viral kinetics data, I think, is a really important thing. Fourth of 5 challenges is figuring out how to incorporate the right modeling frameworks into these tools. So in addition to some of the modeling frameworks that I had mentioned that sort of exist in code bases, there was a lot of sort of public-private partnerships and academic-private partnerships that worked on developing viral kinetics models during the COVID-19 pandemic. I'm highlighting one here, the COVID-19 Pharmacology Resource Center, that was really oriented towards viral kinetics modeling for drug discovery. A super interesting idea, but unfortunately is now defunct. And so again, there's a question of how we make these kinds of modeling efforts sustainable and what the right balance is between mechanistic and phenomenological models, sort of what level of detail we want to include in sort of these out-of-the-box models that we might be able to use to gain insights from the data that's collected. We've gotten a lot of inspiration from modeling efforts for capturing the effective reproduction number. So this is an example of a dashboard called COVID-Estim that took standard surveillance data and applied various types of models to them to give us insights into what the effective reproduction number was doing across US states over time, depicting it both as a map and as a plot over time. And so one could envision these types of sort of flexible models that can be run in browser to give us similar insights about viral kinetics over time during an epidemic. But I think it's an open question how exactly we do that. And the last thing related to that is how do we depict and visualize viral kinetics data? There's been a lot of work on this for epidemiological data, so thinking about outbreaks in the population. How do we do things like represent the vertical scale, and how do we do things like represent uncertainty? How do we compare between different epidemic trajectories? And I think we can rely on a lot of those insights, but it's also not clear which of those will apply well to viral kinetics data visualization and representation, and which of them won't. So I think we need a lot more work on just the display of viral kinetics data to make sure that that something like these efforts, continuous viral kinetic surveillance, has the impact that we hope it does. So to conclude, I do think that continuous viral kinetic surveillance could be a useful addition to existing surveillance efforts. We have a lot of information about viral kinetics in HIV and SARS-CoV-2, but I think that there are a lot of applications for other types of viruses and pathogens, and so there's a lot of room for growth here. Doing this now, sort of in quote unquote peacetime, I think is really useful, both because we can learn things about existing pathogens, but we also can get information that we can use as priors and baselines when emerging infectious diseases come about. This was one of the major gaps during the COVID-19 pandemic, was we didn't even really know where to start with the viral kinetics trajectories for SARS-CoV-2. So having these baselines could be really critical. There are a lot of challenges to overcome that are technological, logistical, and economic, and the list goes on and on. But I do think that if we as a community are able to succeed in incorporating viral kinetics surveillance into our active surveillance efforts, we could have a really big impact on epidemic response, and also potentially answer some really useful and important key questions in basic immunology and epidemiology. So that's it. Again, I want to acknowledge the folks who have been really involved in this work from the beginning. And then listed on the left are people we've worked with over the last couple of years for gathering a lot of the data that I depicted on these slides, and analyzing it and thinking through sort of what the value of viral kinetics data might be. So that's all I have, and I would be very excited to hear from others about sort of what you think about all of this. Thanks so much.
Thank you so much for this amazingly interesting talk. Does anyone have any questions? If so, raise your hand, post them in the chat. I see Renee has a hand up. Go ahead.
Yeah, thank you so much for this fantastic talk. Really, really great work and well presented. I was wondering, how do you see the role of these human challenge studies? So this very unique situation where you can actually uniquely observe even the exposure, and potentially so you could do a serial testing and observe the full time series. Maybe you have that information early on, and you can even maybe learn something about infectiousness and how that relates to viral load. How do you see the role of this in the full picture that you get, and what information is there to gain from those studies?
Yeah, thanks, Renee. I'm very excited about human challenge studies, and I think that they can provide a lot of really useful information for this kind of thing. The two sort of key challenges, I think, the key challenges that face human challenge studies is one, I think that the ethics can be challenging for an emerging pathogen, given that we don't know a lot about it, and so at the moment they're probably best suited for pathogens that we know quite a bit about, and it can be hard to spin up human challenge studies early during an emerging infectious disease outbreak. And sort of the other key challenge is that it seems like the viral kinetics trajectory that a person goes through may depend in really important ways about how they're infected, and specifically the inoculation dose. And so there can be some confounding where it can be difficult to know how to translate challenge studies to natural infections. They do definitely apply, but it's not always a sort of like one-to-one correspondence. Just because in challenge studies, the way that a person is infected is often very different than the way that a person is infected in the general community. But I think that oftentimes those difficulties can be really counterbalanced by the ability to have, a huge amount of control over the study design, the ability to have really good surveillance on when infections happen and maybe secondary infections that they cause. And so I actually think I think that human challenge studies are a great direction to be going here, and would love to see more of them.
Thanks. There's one question in the chat from Sharon who asks, when we're talking about kinetic viral data, does it mean viral load only? And if so, what type of human samples would you need to collect that data?
Yeah, so I have today focused exclusively on viral load over time. But one of the things we're trying to do, and again, not just us, but is to take a step back and sort of think about sort of infection trajectories, infection kinetics more broadly. So you can think about viral load, you can think about the dynamics of like antibody dynamics, and there are all sorts of things that change over the course of the natural history of infection that we might be interested in, and that we might be interested in translating between. I do think so. If I've understood the question correctly, I think your point about what types of samples we need is really important. And part of the reason we focus so much on viral load, and specifically viral load in the nose and in saliva, is because it's relatively straightforward to collect. We might be much more interested in either exhaled virus, if we're interested in transmission, or viral load in the lungs, for example, if we're interested in the clinical severity and course of disease. But measuring virus in the lung is pretty invasive and challenging. And measuring virus in breath, there are tools that are available for that, but they pose challenges as well. And on the other hand, maybe we need blood draws for other types of kinetics that we might be interested in too. So there's really sort of just a logistical balance here, an operational balance that we need to figure out, where we want a good volume of data, and we also want the right data. And figuring out sort of where that balance sits, I think, is a really critical one. But I do think that being able to translate between specifically viral load kinetics and all of the other different types of kinetics that happen over the course of an active infection is a really useful thing to do. I know there's been some recent work by a colleague of mine named Christopher Boyer, who's been thinking about how to build sort of statistical models to do this comparison between different types of kinetics during infection. And so if you're interested, I might be able to point you towards some of that.
Thank you so much. I have a very quick follow-up. I see there's also something in the chat, which is how much is already being done, or are you thinking about linking this especially to serological data? Because it makes sense to me, if you have the kinetics and the serology, that it's kind of, you get exponentially more information. So I was wondering what.
Yeah, absolutely. And I think that the dream would be not just doing this surveillance alongside existing surveillance efforts, but actually sort of like tightly integrating it. So when a person is infected, having their kinetics trajectory and having something about their serology, and making sure that we're doing good baseline serological measurements in the population over time. Of course, with each layer of additional data, there are additional complexities that come in. So some of the work on viral kinetics that we did using the cohort from the NBA also included some serology, and we were able to see some differences in these viral load trajectories for SARS-CoV-2 between people who had sort of high serological titers versus low serological titers at a given point in time. But of course, there then also the what exactly you're measuring serology to is really important, and which variant and these different kinds of things. And so the more data we have on these kinds of things, the better, absolutely. And I think figuring out how to prioritize which data we collect is something that I don't know yet how to do. And I think is really just is really the key question here. How do we make these things sustainable? How do we make these things so that affordable, so that we can do them and get the information that we need. Yeah. So absolutely, super excited about serology and also sort of the time course of serology. And I would love to see more of that happening and integrating that with viral kinetics. So maybe a longer term goal, but yes, something that we definitely want to do.
Thank you so much. That's great to hear. I have a hand up by Irene.
Yes, can you hear me?
Yes,
Yes.
hello.
Yes, hello. It was great hearing you. I enjoyed very much your presentation. I had a question regarding, so when you mentioned regarding the therapeutics, like sort of when will we get the most effectiveness out of giving a treatment if we know more information about viral load? Is there any insights regarding COVID-19 on when you wouldn't give a therapeutic medicine in terms of any viral load, or is that more related to the effectiveness of the therapeutic? I didn't understand quite well that part. Like, are you giving a certain medicine because it will be more effective, or it is actually contraindicated to give that medicine because of the viral load?
Yeah, so I think that, and with full disclosure that I am not a clinician, and so I may well butcher this explanation as well. So clinicians in the audience, if you're squirming in your chairs, please, please correct me. But basically, when giving any kind of antiviral therapeutic, it's sort of a risk-benefit analysis, and for many antivirals, and certainly for things like Tamiflu and Paxlovid, the effectiveness is good when it's administered early in infection, and the effectiveness sort of drops off quickly as the infection continues. And so my understanding is that it's not like you're not necessarily going to actively harm someone by administering a therapeutic too late. But if it's pretty clear that there's not going to be a benefit, and I mean, some of these things do have pretty important side effects.
Yeah.
Then in that point, then maybe really what you're doing is trying to sort of weigh this risk benefit calculation. And so really what we're trying to think about is like, how do we provide clinicians with a little bit more information for conducting that risk benefit analysis for a patient who's sitting there in front of them? Is there something that we can do that might allow them to make more confident decisions than they're currently able to? So I'm definitely not going to stand here and like try to define some kind of treatment algorithm for a given patient. But it does seem like if we could provide some kind of indication for how do you interpret a patient's CT value if it is this high after this many days of symptoms, that might allow us to sort of give clinicians a little bit more confidence in the treatments that they do administer. So that's the idea there.
Okay, great, thank you.
Yeah.
I think we have one final question in the chat, and then we're out of time, which is again by Sharon. Wastewater is a new source of data. Can this also be used for modeling, or is it not detailed enough?
Yeah, so wastewater data is incredible, and I think that this maybe gets at a point that I went over very quickly at the very beginning, which is that you can measure viral kinetics in a lot of different spaces. So I've been thinking about viral kinetics sort of anatomically within the human body at different sort of anatomical sites. One of the challenges with wastewater data is that we're of course sort of like mixing together the virus from potentially a lot of different people, and so of course you can't really get viral kinetics data about an individual person from that. But maybe we aren't always interested in viral kinetics at the anatomical level. You know, one of the things that we've been thinking a little bit about is like, how do we think about viral kinetics in built spaces? So if you think about a classroom or something like that, the viral abundance in that classroom is going to depend on sort of when people come in and out of that classroom. It's going to depend on how many people are infected. And so you can sort of think about viral kinetics at a larger level, about ambient viral kinetics. And I think that wastewater is very much in that same spirit, where I know that at the University of Colorado during the COVID-19 pandemic, there was some wastewater surveillance that was being done at the dormitory level. So we were actually sampling wastewater from individual dorms to try to understand if an outbreak was happening within a specific building. And so I think that things like that, especially more targeted wastewater surveillance that are capturing things at the neighborhood or even the building or household level, again, continue to raise really important ethical concerns, but also can start to give us a little bit more information about viral kinetics, both at the individual level, but then maybe also at larger scales that we might be interested in when we're thinking about the spread of disease. So I think it's maybe not quite our understanding of wastewater and how to normalize for all of the different fluctuations that can happen there is not quite robust enough to directly inform viral kinetics models at this point in time, as far as I'm aware. But I think it's a really exciting area for work going on.
Awesome. Thank you so much for that amazing talk and a great discussion. And thank everyone for coming.